All right, so thanks. So as I was introduced, I'm a professor at the University of Luxembourg, but I've been working with BXDs and multiomics, especially for the past about 10 years. So today my talk was nominally about metagenome and metatranscriptome, but what I'm really interested in is trying to see how molecular profiles change across tissues, across omics layers, across time. So metagenome, metatranscriptome is relatively recent that we can acquire good data. And now what I'm interested in seeing is how does essentially how all these different tissues and data sets, how do you put them together to come up with some sort of cohesive picture of what's going on physiologically? So if I can advance the slide, there we go. That's so, see this close. Can I use this? Yeah, how does this work? So this works. All right, there we go. That's better. So what I've been working on about the last 10 years was a BXD aging project we set up in Memphis in 2012. The bulk of it finished in about 2019, where we aged about 2,200 mice from 90 different BXD strains, half chow diet, half high-fat diet. We let about 2/3 of them live out their natural life, and the other third we sacrificed over various times so that we could get the biobank of the molecular changes as a mouse goes from about 6 to 24 months of age. So this was published a couple of years ago. And what I've been working on since about 2019, when we finished the bulk of this project physiologically, was to take different tissues and extract different levels of molecular data. So looking at the metabolome, the proteome, the transcriptome, the metagenome, and trying to see if we make these profiles, for instance, initially in the liver was the first tissue I focused on that was published a couple of years ago. So if we study 291 of these mice with their transcriptome in the liver, 315, all of these overlapping in the proteome, et cetera, with the metabolome. Basically, what can we figure out about liver physiology? This is relatively straightforward, but we've been trying to work on since then is taking this across tissue as well. So for instance, looking at the transcript expression in the cecum, which we've done on 79 animals, looking at the microbiome, so the contents of the cecum in another 210 animals, and again, the same 79 animals with the metatranscriptome, and then also the white adipose tissue with the proteome. And essentially, we can analyze these all separately. There are a whole bunch of different layers of data, but how do we actually come up with some sort of cohesive picture of what's going on physiologically, rather than just having a bunch of different data sets that have been done coincidentally in the same mice? So first off, what I was interested in seeing is just how do these molecular networks vary across tissue? So I have this study published a couple of years ago in liver multiomics. Okay, this has been done by a few groups, Steve Gigi, Gary Churchill. There are other groups that have done a fair amount of this now. We have a fairly good idea of how mediocre the association is between transcripts and proteomes. But what if we look across tissues? Of course, people have done this at the transcript level for a long time. And more recently, people have done this at the protein level. And then basically, how consistent do we predict an outcome like the age of a mouse or the obesity of a mouse if we're using molecular profiles from these different tissues? So if you look at something relatively straightforward, so if you look in this case, this is the effect of a high-fat diet on protein expression in the white adipose tissue in the liver. So it's about the same number of animals, relatively similar sample sizes in terms of the proteins there. We see that's a relatively similar picture. So there are a few more significant results in the white adipose tissue. The fold changes are a little bit wider as well. But broadly, we see a similar response for the effect of high-fat diet on protein expression. But if we want to look at any of these particular proteins, so I've highlighted a few that you can see overlap, like ApoA4, but actually the bulk of these don't really overlap. So the correlation coefficient between the average effect of high-fat or chow diet on the expression of proteins for the 11,000 peptides that are expressed in both of these tissues, it's a pretty mediocre 0.33. It's highly significant because the sample size is huge. But if you wanted to say, I'm going to take a particular gene and make a hypothesis and make a knockout of this, well, 0.33 is not really good if you're going to spend a year and a half and $20,000 validating that gene. So this is something that people have also showed now for a number of years, and I don't think this is controversial anymore, but it was at the time, is if you look at the effect within a single tissue, so in this case, just within liver, if you look at the effect of high-fat diet on the transcriptome of the liver versus the proteome of the liver, again, you get a pretty mediocre correlation. It's okay, again, the sample size is huge. If you see 0.42 and a p-value of minus 159, you're relatively happy. But if you wanted to use this to actually pick a particular hypothesis to try to validate, then you're going to be a little bit less sure that you are going to not make your PhD student very sad when their project doesn't work after 2 years. So sometimes you actually do get consistency. So the vast majority of results they're not very consistent across tissue, and especially not across tissue and across omics layer. Occasionally you get lucky and you get something that you're really happy with. So in this case, celluloplasmid actually associated very well with age. There are also a fair number of such proteins and transcripts that correlate across tissue with obesity or with diabetes status. So you certainly can find consistent results like this. This was the only one that was super convincing that worked across tissue and across chow and high-fat diet. So for whatever reason, celluloplasmin associates very well. It's a good predictor of age in the BXDs, not lifespan, but their current age. So this consistency or inconsistency, you want consistency if you want to pick a particular gene that you want to knock out or if you want to modify and study, but the inconsistency between omics layers can also be useful as well. I hear sometimes people say that now that proteomics has gotten pretty good, maybe we should basically abandon transcriptomics because the protein is what's actually working. And you can say that, but it's not really quite right, because the inconsistency, it's like saying, well, you get great results in the liver, so let's not measure other tissues. The liver is such a good tissue. Why would anyone bother with the pancreas? It's hard to do mRNA in a pancreas. Just forget about it. So it's the same also if you look at in this case, this is oxidative phosphorylation in the liver from the same exact animals at the same exact time at the mRNA level and the protein level. And just generally what you can see here is this is the 5 major components of the electron transport chain. At the mRNA level, you don't see any association between the colors. They're all kind of co-transcribed together. But if you look at the protein level, you see that there's some sort of structural association. You see that each element of the electron transport chain, here's complex 1, here's complex 4, here's complex 5. And you get additional information about what's going on. And what's a little bit less clear here is the transcript level, you can actually figure out which of these genes are mitochondria. You can see if this transcript is coming from the mitochondrial genome or from the nuclear genome, which you can't see at all from the protein level. So you're getting good information from both of these cases. But of course, if I didn't already know what OXPHOS looked like, it would be hard to actually form this if I had no sort of ground truth to start with. So what I've been working now is something that's even more complicated because we have so little ground truth with it, which is the microbiome. So we can now take something like the cecum, we can homogenize it, we can get the metagenome, we can get the transcriptome, we get the metatranscriptome. It's now relatively cheap to do this, and the informatics tools have gotten a lot better. So we can develop all of this data, but now the question is, what do we align it to? What do we actually make out of this? So there is some information. People have been doing 16S for 10 or so years now. And you can basically make sure that, yes, your data are on the right track. We can see high-fat diet decreases diversity of the microbiome. One thing that was a little bit of a surprise to us is here on the x-axis, this is the age of the animals. The age of the animals didn't really have any impact on the diversity of the microbiome. The effect of diet and the effect of genotype, which is the difference between the dots within a diet, this vastly overpowered any estimate or any factor that we saw that was linked to the age. We also didn't really see any effect of the maternal imprinting, at least not by the 6-month time point we were looking at here. Basically, everything was a rounding error compared to the effect of genotype or the effect of diet. So if you look at this a little bit more carefully now, so now that we have really whole genome sequencing, we can do 10 million reads of microbiome metagenome. We can look at the genus level, or we can even look at species level of these data sets and try and figure out, well, which parts of the microbiome are actually useful and what is this actual connection between the gut-brain axis more generally. So if we zoom in, that's the same figure. If you look at this case, this is the metagenome from 200 animals in the chow and high-fat diet. We can see some pretty obvious patterns here, like Duncaniella, the bottom purple graph. It goes way down in high-fat diet compared to chow diet, and Lactobacillus the opposite. The challenge now is if we look at the metatranscriptome, we see a very different picture. So in this case, the Duncaniella that was so striking in the chow diet is now almost a rounding error, and it's almost completely eliminated the high-fat diet. And conversely, if you look at the Lactobacillus, it's sort of a different picture where it's pretty highly abundant in the high-fat diet metagenome, but it's extremely abundant. Sometimes more than half of all metatranscripts come from the same genus across all of these samples. So if you look at this in a different way, sometimes it's very consistent as a factor of diet. So with the Duncaniella and Lactobacillus, we see it's rather consistent that the metagenome is being changed versus the metatranscriptome as a function of diet. But sometimes like this Heminiphilus, the blue line, in this case, we see that there must be some sort of genetic effect that's vastly outpowering the effect of the diet. So in this case, some of the mice have a lot of it, other mice have very little of it. It actually tends to cluster very well by the genetic background, although it doesn't actually map to a QTL. So again, we can look at this same thing at the high-fat diet, and we see sort of similar patterns, but it makes us a little bit hard to say, well, if we see this complete elimination of Hemiphenus or Lactobacillus between these mice, how do we actually use that to figure out something physiologically relevant? So that's what we're trying to do now. First off, I want to know how consistent are these results. So in this case, this is the metagenome of Duncaniella and Lactococcus across all 200 animals. If you look at the metatranscriptome from the subset of 80 of those same animals, we see it's, again, diminished or increased in the same directionality. The actual y-axis changes a lot. So Lactococcus is basically a rounding error in the metatranscriptome, whereas in the metagenome, there's quite a bit of Lactococcus in the chow diet animals. But what's nice to see is actually these correlate pretty well. So even though the actual magnitude is vastly varying, of course, these are percentages and they're not absolute counts, they actually correlate quite well. And if you look across all 187 taxa that we measured at both the metagenome and metatranscriptome level, the correlation coefficient is actually super high on average. The general correlation between the metagenome and metatranscriptome is like 0.7 or 0.8. So at least it's very consistent, even though we're seeing these large changes in abundances, which again, because they're normalized, it might be a little bit misleading. So now what I'm trying to do, and I haven't an answer to this, is try to see how does this microbiome actually associate with all this gene expression data. So in this case, we can see how does Oscillospiraceae correlate with the gene expression of the liver, the white adipose, everything else we've measured. So in this case, the yellow line or the orange line in high-fat diet, there's absolutely nothing significant at all. In the chow diet, there is this significant associations, basically the difference in area under the curve of the dotted between the dashed line and the solid line. That's the number of significant associations we're seeing, more than expected by chance. But of course, the correlation coefficient there is extremely low. It's more than you'd expect by chance, but it's also such a tiny amount of variation explained that it's hard to actually make use of it if we see that these are correlation plots like this, where we have Duncaniella versus SQRDL. Well, okay, it only correlates in chow diet with a relatively mediocre nominal p-value. It would be okay if we had a prior hypothesis, but if we're trying to find this completely de novo, then something like this Bacteroidetes versus Jun-B, well, it's a little bit hard to be excited about these nominal p-values, even though you're seeing them in both of these diets. So again, this is something that we're having trouble with now is, yes, we can do gene mapping. Yes, we see that there's clear genetic association between certain species of the microbiome or certain genre. But if we're not seeing it consistently, and we don't usually see it consistently, then how do we actually make use of it and ensure myself that if I actually want to do Bacteroides and figure out what is causing one of these QTLs, well, if I only see it in my study and I don't see it in another BXD study or even another mouse study, then how am I going to actually nail down this causal gene if it's due to an environmental factor that's so difficult to identify? So now we have all these results that correspond with what's known from 16S. So the firmicutes to Bacteroides ratio is something that's very well known to change in high-fat diet. It goes significantly up in high-fat diet, which is the case here. It about doubles in the BXDs, but there's a large variation from mouse to mouse. Some of these animals have a 10 or 12 or 15 times difference, where others are more like 2 or 3 times. So there's a clear genetic effect, but then we need to figure out how consistent is this and how do we actually solve it. So this has largely been the PhD project of Ziyun Zhou. So we've been working on this about 2 years since we got our samples and everything set up in Luxembourg. And a big thanks also to Paul Wilmes and Rashi Halder, who are big microbiome people in Luxembourg who helped us get on track. Liver proteome was me when I was in Zurich. The white adipose proteome is Yibo Wu, and it's not published yet, but we're working on that now. And then the mouse cohort, you probably all know these people. So that's where it came from initially. So I guess we have a little bit of time.